This is Unit 14, Human Immunity by Joshua Evans, Section 1. The first line of defense are primarily physical and chemical barriers that prevent entry to pathogens. The second being non-specific responses from your immune system upon detecting foreign particles. The first line of defense, your primary physical barrier is skin. It physically stops pathogens from entering. Second is your mucous membranes. This is where, because they're very sticky, they physically stop the pathogens from being able to move, essentially. And then you have the urinary system, which flushes them out using urine. A response to injury or infection is inflammation. This is where your blood vessels dilate. It increases blood flow and allows more immune response cells to flood to an infection zone. Immune cells congregate around here, adding to inflammation, allowing a large supply of proteins which fuel the immune response, and the body temperature increases, which is better since many pathogens struggle with high temperatures. Phagocytis is when a immune cell essentially surrounds and breaks down foreign material or pathogens. You see is it detecting foreign material. It then forms a vacuole to engulf it. Once it's inside it, it fuses, and then the foreign material is broken down and expelled. Then there's the complement system. The complements are plasma proteins which bind and cause cascade reactions. These proteins react with one another to cause microbes to undergo three main outcomes: phagocytosis for your macrophages, destruction by leukocytes, and osmotic lysis of bacteria, which is essentially when they cause them to burst. Section 2, the third line of defense and autoimmunity. The third line of defense is the point at which the immune system has adapted to the pathogen and begins using specific methods to destroy it. The main two roles of the third line is to identify and destroy specific pathogens and to build the immunity against pathogens for future infections, such as like your memory. First off, antigens, molecules including protein, carbohydrate, and nucleic acid, and fat. These antigens make your body aware that there is a pathogen invading and begin the immune response. Pathogens often use antigens to infect the body. Example being influenza requires a protein to infect. The body is constantly trying to recognize these antigens to start an immune response. Acquiring immunity. Immunity isn't inherited in the body and must be learnt. A newborn baby only has immunity to very few things. So it's something you have to develop throughout your life. Immunity can also be given artificially via exposure to vaccines or passively being introduced with preformed antibodies into the body to generate a protective immune response. The immune cells themselves, the white blood cells, also known as leukocytes, are the most important part of the immune system, as they make up the majority of it. These cells are not a single cell, but in fact 5 separate types of blood cell, all which are made in the bone marrow. First off is the neutrophils. This is the most common form. They're medium size for an immune cell. They have multiple functions. These cells tend to cling to the side of blood vessels to block access from germs. They also are the first white blood cells to react, and they perform phagocytosis, where the engulfing I referred to earlier. Lymphocytes, these are small round cells with large nucleus. They have humoral immune system, humoral immune system, which is involved in antibody production. It's found in the lymphatic tissues such as spleen, tonsils, and lymph nodes. Lymphocytes B produce antibodies and make memory cells so that the specific pathogen can be destroyed faster the next time it enters the body once it's identified. Monocytes. These are the largest of the white blood cells. Monocytes turn into macrophages when they exit the bloodstream. As macrophages perform phagocytosis on dead cells. Using their large size, they can ingest large debris, and in a wound, they are unlike any other white blood cell. Eosinophils. These cells release toxins from their large granules. These toxins are especially good at attacking worm infections and allergies, and you detect these in people who associate high with allergic reactions. Basophils. These are the rarest of white blood cells. They expel anticoagulants and antibodies against hypersensitivity in the bloodstream. They react very quickly to pathogens, and they contain histamine, which dilates blood vessels, allowing more immune cells to reach the infected zone quickly. Acquired immunity. Many cells in the immune system are B cells and T cells. These are called lymphocytes. There's a third type called the natural killer cells, but however they're part of the innate body response, not this. The next slides will refer to the types of T cells. Helper T cells, these are most common. Their function is to aid the immune system in a multitude of ways and work as a regulator for virtually all immune functions. They mostly release lymphokines to control the rest of the immune system. They also assist in helping B cells grow and mature in their active form. Cytotoxin cells. These cells work against viruses and cancer cells / tumors. They resist the ability of foreign pathogens by grafting themselves onto our body cells. They use activated APCs and helper T cells. The cytotoxin T cell can produce cytotoxin proteins, as for their name, which rip holes in foreign organisms, causing the enzyme can release enzymes capable of destroying the cell structure. This cell often attaches itself to an intruder cell, and it releases its toxins directly into it, causing a localized destruction of that cell and causing, say, the virus not to be able to replicate in this manner. Suppressor T cells. These T cells can suppress the functions of both helper and cytotoxin T cells. They do this to regulate other cells in the immune system, potentially stopping them from causing more damage to the body than is necessary from an infection. This makes them very important when it comes to autoimmune attacks, as it's essentially stopping your own body from attacking itself. B cells. These are created in your bone marrow. Once active, they release mass amounts of antibodies, kind of flooding the infection zone. Once antibodies production, B cells present antigens to T cells and provide signals for T cell activation. Plasma B cells produce and secrete only one type of antibody. These antibodies are secreted directly into the circulatory system. Immunity is maintained by the continuous release of these antibodies. Memory B cells are formed after stimulation. Wherever there is an active infection, these then retreat to lymph nodes where they remain dormant until activation for another infection. Immunoglobulins, or your antibodies. Antibodies have a variety of roles in the immune response, targeting infection, recruiting immune cells, and neutralizing toxins. The structures of immunoglobulins tend to be made up of 4 chains. These 2 chains are normally denser, giving it effects of 2 heavy chains and 2 light ones. There are several types. Antibodies G. The most common immunoglobulin is one of the main activators for complement proteins. They can attack any foreign material it recognizes, and the only type of antibody that is transferred from the mother to the fetus. Type A. This is the second most common. It defends the soft, moist mucous surfaces of the body, such as within the mouth. Low amounts of this can cause infections in the mouth, throat, and lungs. Type M. The structure of these are clearly different from the rest. It comprises of 5 normal ones joined at the base. This is the first to be produced in any antibody response. The largest type is type E. It indicates in the healthy body it has low levels. These levels drastically increase as a response to parasitic infections and allergic reactions. Hay fever is caused by high levels of type E. And then there's type D. Its function is largely unknown. All we know is that it's related to antigen receptor on the surface of B cells. Getting to the next point, which is recognizing self and the non-self. Our cells display specific antigens, which the immune system will then designate as self. The levels of response from the immune system is not consistent and depends on the level of danger that the immune system believes the non-self presents. This is determined on how similar the other is to the human's antigens. So starting with MHC class 1. These are expressed on the surface of all nucleated cells. They act as an early warning system. If the cell that MHC1 are attached to contains foreign proteins, then MHC1 will degrade these proteins into fragments and display them on the cell surface, making them recognizable to the immune system. MHC class 2 are similar, however, they are only on the surface of certain cells. These act as extracellular presentation, exposing extracellular pathogens such as bacteria. These pathogens are endocytized by B cells and phagocytes, Digested and then antigen fragments are presented by the MCH2 molecules on the surface of the immune cell. T cells then detect this and target cells expressing the same antigen. Tolerance to yourself. As the immune system generates this massive array of immune cells, it constantly culls those that are self-reacting or malformed. If the immune system fails to remove these rogue cells, then they may activate B and T cells against the immune system's antigens. This is the basis of all autoimmune diseases. However, no one knows exactly how the tolerance itself fails. Rejection of transplanted organs. Transplanted organ is a foreign material, as that's the nature of it, so it can cause the immune system to activate. The antigens on the cells of the organ are different, causing them to be identified as non-self. The solution to this is to match the recipient to the donor as closely as possible, but this only reduces the chance of rejection, not eliminate it completely. Tissue typing can be used to make sure that the organ is similar as the host, and monoclonal antibodies can be used during and after surgery to knock out the specific T cells which will cause rejection. Types of rejection. Hyperacute rejection occurs only a few minutes after a transplantation or transfusion, and this is when antigens are completely unmatched. Some sort of massive medical issue would occur for this to happen, and if a tissue is removed right away, the recipient will die. This also occurs in recipients if they're given the wrong blood type. Acute rejection. This occurs any time after the first week from transplant to possibly 3 months afterwards, and everyone receives some form of it. The chronic rejection happens many years after transplant, or from transplant to many years after, where the body's immune response constantly is breaking down against the damage in the new transplanted tissue or organ. Some examples of this. Arthritis is autoimmune disease and inflammatory disease. This means that your immune system attacks your healthy cells, causing inflammation in the affected area. This disease primarily targets the joints, including the hands, wrists, and knees, causing the hands to bend in horrible ways. And then there's multiple sclerosis, which is affecting both the spine and the brain. Exactly what it says there. The immune response specifically attacks the myelin sheath, which protects the nerves of these two areas, or the sort of nervous system. The symptoms can include fatigue, difficulty walking, numbness, issues of balance, coordination, and problems of thinking, learning, planning, and etc. The list of symptoms is massive. Section 3. Acquired immunity and vaccination. Acquired immunity can be described as using these words: active or passive, where immunity was gained from exposure to a threat; active, or antibodies have been introduced to a person without exposure to the threat; passive. Natural or artificial, whether immunity was gained as a natural part of living or was induced via artificial medical procedures such as vaccination. Active acquired immunity. Natural active acquired immunity. If a person is exposed to a pathogen from the environment, the immune system makes memory B cells and T cells remember the pathogen so that it can respond quicker if it's ever exposed by the same thing. Artificial active acquired immunity. Artificial active acquired immunity is essentially the same process except that the pathogen is introduced to the body artificially. This is done via vaccinations. Vaccinations are safer as the pathogens can be inactive or dead, or sort of like neutered. There is also a lower severity of illness. Passive acquired immunity. Natural passive acquired immunity occurs when antibodies are transferred from one person to another, such as from a mother to a fetus, as I referred to earlier in this presentation. Artificial active acquired immunity. Artificially induced passive immunity can come from giving a person an injection or a transfusion of antibodies from another person. This effect doesn't last very long as antibodies gradually break down and are not replaced by the body, as it doesn't know how to make them. The immune response to vaccination. Vaccinations can contain live pathogens specifically treated to make them harmless. Harmless fragments of pathogen or toxins and death pathogens. These are injected into the body to get to the immune system to treat it and for it to generate a response against it. If the same pathogen is again introduced to the immune system, then the white blood cells will be able to rapidly deploy and destroy it. This is referred to as immunological memory. Herd immunity. Some people are too vulnerable for vaccination, such as those who have weak immune systems, that being the old and the very young. Since many diseases are contagious, it is important to reduce the likelihood of being exposed to pathogen and by doing this we vaccinate a large portion of the population, making it much harder for this disease to spread. The critical mass that is required for herd immunity varies between diseases. Many of them are between like 75 to 80%. Diseases such as smallpox are destroyed completely via this process, same as polio. vaccination programs. In the UK, the NHS has many local clinics and pharmacists that can help adults fit vaccination into their schedules and allow the vulnerable and disabled to be able to be vaccinated without much travel. Children are also vaccinated throughout their childhood to protect them from disease. These are not normally done via visits or health visits and nurses and a GP. They come to schools as well to vaccinate children. The factors affecting vaccine uptake. The season. Some diseases such as the flu are more common in certain seasons. People are more likely to get vaccinated when risk is higher. Social upheaval. The spread of misinformation by, say, groups like anti-vaxxers can persuade people against vaccination. While these great groups use pseudoscience and poor sources, people with less of an understanding about vaccines can be dissuaded. Illnesses and allergies. People may be unable to take vaccinations such as they have allergies to from the components and their weak immune systems or are currently ill. Availability. Vaccinations may be difficult to distribute in areas of poor infrastructure and funding. Benefits of vaccination. Vaccination has eradicated many dangerous diseases such as polio, which can destroy a person's life and rob them of their movement. It massively reduces child mortality rates. It reduces the pressure on the medical services for taking care of these people who would be ill, and reduces the development of antibiotic resistance in people, which is a humongous problem, and also encourages economic growth as more of the population can work and aren't bogged down by medical issues. This has been Unit 14 Human Immunity by Joshua Evans. Thank you for listening.